Good morning, everyone. Now we discuss about Salmonella typhi. It is one of the bacteria which cause the infection typhoid fever. Now we want to know the overview of typhoid fever. It is a life-threatening infection caused by bacterium Salmonella typhi. It is usually spread through contaminated food and water. Once the Salmonella typhi bacterium are ingested through contaminated food and water, they can multiply and spread into the bloodstream. First of all, the organisms once enter into the bloodstream, it will cause very serious issues to human being. Then, the urbanization and the climate change have the potential to increase the global burden of typhoid. Some key facts about Salmonella. As of 2019, estimated 9 million people get sick from typhoid and 110,000 people die from it every year. Some of the symptoms which includes prolonged fever, fatigue, headache, nausea, abdominal pain and the constipations are diarrhea. Some patients may have their rashes, sometimes it will happen. Severe cases may lead to serious complications or even death. Next we enter into the introduction. Salmonella typhi caused the fever called a typhoid fever. It is also called as enteric fever. It is prospectively multisystematic illness that has been public health problem, especially in the developing world, which is caused by Salmonella typhi and paratyphi. The enteric fever is a cumulative terms, both typhoid and paratyphoid fever. In 1880, German pathologist Carl Joseph Ebert discovered the microbes that cause typhoid fever. Next slide shows morphology of typhoid fever, which is caused by Salmonella typhi. In this Salmonella typhi are gram-negative bacteria belongs to family Enterobacteriaceae. Non-spore forming, non-acid forced, non-capsulated. The locomotor, it is a motile organism. The locomotory organelles having peritracheous flagella, which is the peritracheal flagella means the flagella found around the cell wall. Size varies from 2 to 4 in zero point 0.6 micrometer. In this organisms appear facultative anaerobes. The facultative means both aerobic and anaerobic condition may be possible with respiratory with the fermentative metabolisms. In case of biochemical test, this organism possesses oxidase positive and catalase negative. Next, this is the structure of Salmonella typhi. The peritracheous flagella, which possesses flagella around the cell wall. Systematic classification means domain. In this organism, domain is bacteria, order Enterobacterial, race family Enterobacteriaceae and genus Salmonella. The species is typhi or paratyphi. Cultural characteristics. The optimum temperature for this organism for growth at 37 degrees Celsius and pH 6 to 8. They can grow in ordinary media like nutrient agar, McConkey, Wilson, Blair, bismuth sulfide agar, XLG and deoxycholate citrate agar. And McConkey agar, in this organism produce small circular translucent NLF colonies. The NLF means lactose fermenting colonies. Because the McConkey agar is also called as a differential media. It may be lactose fermenting or non-lactose fermenting. The Salmonella typhi comes under the category of non-lactose fermenting organisms. In another way, Wilson and Blair bismuth sulfide medium, the organisms and the colonies are appear jet black with the metallic sheen colonies. In SS agar, black centered colonies are observed. XLD agar, slight pink colonies with the black centered are observed, and deoxycholate citrate agar, black colonies are formed. In this organism, they possess three major antigens. First one is HR flagellar antigen, OR somatic antigen, and VA antigen. The HR flagellar antigens more serious antigen which may cause Salmonella typhi and paratyphi. The bacterium surface are covered with lipopolysaccharides because this organisms possess gram negative organism, so that the gram negative bacterial cell wall contain lipopolysaccharides. The outermost portion of LPS is the O antigen. Flagella wipe-like tiles that bacteria use to move around. Flagella is a whole structures, while the slender thread-like portions of flagella is also called as a H antigen. Next, we enter the pathogenesis. Both the organisms like typhi and paratyphi primarily cause infections to human. Man's acquire infections. How it's acquire the infections? Via oral route of indigestion of contaminated water and a contaminated food. In this fever may caused by ingestion of small number of bacilli. Typhoid fever can caused by indigestion of small number of bacilli, but the paratyphoid fever needs the consumption of large number of bacilli to produce infections. So normally duration of those infections varies from 7 to 14 days for typhoid fever. In case of paratyphoid, it will take 4 to 5 days. Typhoid fever is more severe than paratyphoid. Next, the major cause morbidity and mortality depends on the typhoid or paratyphoid. On reaching the gut via contaminated food and water, the bacilli undergoes through the following stages. In this bacilli, three stages we have to follow. One is incubation phase, second one is septicemia, third one is localization. What is incubation phase? Incubation phase take the time duration starts from 10 to 14 days. When reaches the bacilli in abdomen, most of them will be destroyed and some of them revived and attaches to the epithelial cells of the villi. These cells then enter into the mesenteric lymph node and multiplications will occur. After multiplication, the bacteria enters to the bloodstream and cause primary infections. So, incubation space will cause a primary infection. Second space, septicemia space. During this stage, the bacilli are entered into the gallbladder, spleen, liver, bone marrow, lungs, kidney, and they can multiplication will occur. After multiplication, it enters into blood and cause secondary multiplications. During this time, they produce toxin and cause fever and other symptoms. The third stage, that means to last space, localization space. The localizations which is localized in a organ like liver, spleen, kidney, gallbladder, etc. and producing the toxins. So that the tissue becomes inflamed, undergo necrosis and result in typhoid ulcer. Later, this ulcer leads to hemorrhage and perforation. In this slide shows the pathogenesis of Salmonella, how the bacteria enter into the gastrointestinal tract, how they are entered and penetrate to the lumen, how they can multiply, and how they are secretion of water and electrolytes, and how they are developed and how they are reproduce the bacterial cells into the localized organs or bloodstreams, etc. etc. Septicemia. What do you mean by septicemia? It is mainly caused by Salmonella typhi, which is very serious cases. Infectious occurs through oral route of bacilli enter into the bloodstream. That is called as septicemia. The septic portions which is caused to bloodstream. Growth and multiplication occurs in different body organs and cause pneumonia, osteomyelitis, primary pulmonary abscess, endocarditis, and meningitis. These are the septicemia. These are the stages which is occurs in a septicemia space, because the particular organism will enter into the bloodstream and the localized organs also affected, and it will cause many serious problems, complications will occur. Next, Salmonella endocolitis. Colitis. It is also known as Salmonella food poisoning or gastroenteritis. It will take the incubation period 12 to 24 hours. After that, the symptoms will develop. Features like vomiting, fever, abdominal pain, and diarrhea. These are the endocolitics, colitics, causing symptoms. Clinical manifestation. Some of the clinical manifestation like fever, rashes, headache, vomiting, pain in the abdomen, diarrhea, bleeding from the nose, dry cough, enlarged liver and spleen, relatively lower pulse rate. These are the clinical manifestations which is occur in the stage of typhoid and paratyphoid. What are the complications? Bleeding time, bleeding from intestine, perforation of the intestine, kidney failure, enteric encephalopathy, meningitis, loss of weight, mental confusions. The typhoid fever requires immediate diagnosis and treatment. How they are transmit to other persons. That is epidemiology. Salmonella typhi is maintained in nature by human carrier. There are three main types of typhoid carriers. One is convulsion, temporary, and chronic carriers. It is mostly transmitted through fecal oral route. Human is the only natural host. It can occur at any time during the year, but it is common during July to September. Diagnostic methods. First diagnostic method hematological investigation. Second one is bacteriological investigation. In case of bacteriological investigation, we need some of the samples, cultures. One is blood, slaughter, stools, urine, duodenal juice. These are the cultures we need to perform the bacteriological investigation. Simultaneously, we do the biochemical test and the slide agglutination like viral test and serological test also doing for diagnosis of Salmonella typhi and paratyphi. Next, we enter how the samples are collected. Blood or bone marrow or clot, stool, urine or clinical sample. These are the samples which is collected in laboratory level. Isolation of the organism, blood culture. In case of blood culture, the organism can be best isolated during first 7 to 10 days of illness. But in at least half of the case, the organism can still be isolated in a second and third weeks. Slot culture in the sense, the slot culture has the following advantages over blood culture. Serum is available for antibody titration. Special cultured bottle are not required, so it is useful under field conditions. Next, culture of feces. In at least 50% of the cases, culture of feces is positive in the first week. So, fecal culture should be attempted in the first week of illness itself. The second thing of the isolation of Salmonella typhi greatly increase in second and third week. Rectal swab give inferior result compared to fecal culture. The second sample, second not only second is another culture sample urine. Urine culture is positive in a quarter to 1 to 3 cases, but there seems to be no regular excretion pattern in the urine. Bacteriuria is usually limited to the first few weeks of the illness, and in great majority of the patient, the urine is free from organism weeks before the feces become negative. Next, serological test. The serological test nothing but the serum sample we are all used. So nothing but detection of antibodies. A large number of serological tests have been devised to detect and titrate the antibodies against the common agents of enteric fever. The tube agglutination test is nothing but Vidal test is a great historical but little diagnostic importance unless fourfold rise in exhibited in two samples drawn at interval of 10 to 14 days from the start of the illness. It may include Vidal test and ELISA. Both are agglutination. Both are the basic principles of agglutination test. In case of Vidal test, in this test, the patient's serum is tested by tube agglutination test for its titer of antibodies against H2O, and V1 suspension of enteric fever bacteria. In case already we told in this organism having the O antigen and H antigen, AH and BH for paratyphi, O1H typhi and AH and BH of paratyphi. Negative control, NC, which means negative control, and PC means positive control. In this test, in case O1H agglutinate means Salmonella typhi positive, AH and BH, which means Salmonella paratyphi will positive result will appear. In case of simultaneously, the PC control also have the agglutination reactions. How to prevent this infections? Sanitary measures is very important, and simultaneously we have to consume vaccines, prefer vaccines. Are the two main ways to prevent enteric fever. Sanitary measures include provisions of safe drinking water. First thing. Second thing, safe disposal of human waste. The third thing is maintenance of high standard of food hygiene. The last one is care in detecting, monitoring, and treating chronic carriers. Next slide shows individual protection measures includes drinking boiled water in iodinated and chlorinated water, care in consumption of food prepared outside, immunization with the typhoid vaccine, avoidance of raw and partially cooked eggs, washing hand with soap and water after using toilet and before preparing or handling foods. People who have recovered from fever to use separate towel till they get completely cured. These are all the individual protection measures. Next, vaccination. What are all the vaccinated method preferred for this infection? Three main types of vaccines are available. One is older heat inactivated phenol preserved whole cell vaccine. Second one is new V1 polysaccharide vaccine. Third one oral typhoid vaccine. These three kinds of vaccines we are administered. Treatment in the way of what kind of antibodies we have to take it away. Take it for the treatment of this infections. Chloramphenicol, ampicillin, amoxicillin, uh, ciprofloxacin, etc. These are the antibiotics which is treated the salmonella typhi and paratyphi like a typhoid fever. Finally, we enter the thank you slide. Thank you. This is the overall view of the salmonella typhi and paratyphi, how they are causing infection to human being, and what are the complications, how they are recovered from this infection by the way of vaccinations and any antibiotics, etc. Thank you, one and all.